Hey everybody, so this lecture is going to be about the endocrine system. So you can see in the diagram here, it's just got some organs and glands labeled, and all of those that are labeled on here are a part of the endocrine system. So what is the endocrine system? The endocrine system is made up of a bunch of different glands and organs that are kind of scattered throughout the body. Those glands release molecules into the bloodstream. Those molecules are called hormones, and that allows them to travel to a target site. So they really can travel anywhere in the body. A target site is anywhere in the body that has a receptor for that particular molecule or for that particular hormone. So when it gets to the target site, the molecule is going to act as a ligand. You'll see the word ligand a lot. Basically, kind of a simple definition for a ligand is just something that binds to something else. So sometimes people will use the term hormone and ligand interchangeably, because basically what's going to happen is the hormone is going to get released from something in the body. Let's say the brain releases a hormone, and that hormone is going to travel, let's say for example, down to the ovaries. So you might hear it worded as the hormone is going to bind to the receptor, or you might also hear the ligand is going to bind to the receptor. So the ligand or the hormone will bind to the receptor at the target site. The target site is just any area in the body that has a receptor for that particular molecule. Now, once the hormone and the receptor interact together, that's going to initiate some kind of change, some kind of biochemical or metabolic change. And so basically, the overall idea of this is that you can have a molecule being released from one area of the body that causes biochemical changes or metabolic changes in a completely different area of the body. So it's a communication system and a control system. So that's the endocrine system, but we've talked previously in 2401 about another control system. You think about that, what that might be. So the nervous system is the other kind of communication control system that we have in the body. So the nervous system and the endocrine system both basically release molecules that control or communicate with other cells in the body, but there's some pretty key differences between the two systems. So let's look at those, starting with the nervous system. So the nervous system is going to release its ligands, which are referred to as neurotransmitters, and it releases them through those action potentials. So hopefully some of this sounds familiar from 2401. You have your neuron with the cell body. It synthesizes neurotransmitters. They get stored down here in axon terminals or synaptic knobs. And then you get this action potential or electrical impulse that comes down, and it causes the release of the neurotransmitters. So key features in the nervous system, notice that the target cell is right there adjacent to the neuron, where the neurotransmitter is going to come out. Also in that area are molecules that can degrade the neurotransmitter. So the nervous system, yes, it will release a molecule that communicates to another cell, but that target cell is right there next to it, which means it's a very local response. You also have things in the area that will degrade the neurotransmitter almost as soon as it comes out. It has about a second or so to act, and then it's going to get degraded, which means it's a very rapid signal and a very transient signal, meaning it's going to happen and it's going to go away. If you contrast that to the endocrine system, what you're looking at here is some endocrine gland, and remember the endocrine system is going to release hormones, not neurotransmitters. So the definition of a hormone is actually a molecule released from an endocrine gland that travels through the bloodstream. So it's getting released into the bloodstream. Now what that does is it allows all of the body, or all of the vascular tissue, any tissue in the body that has blood supply, now has access to these hormones. So it can travel anywhere. So as opposed to the nervous system that was a very localized response, the endocrine system is going to be a more widespread response. It can go anywhere. These hormones are also quite stable, so there's nothing there that's going to degrade them or make them go away. So it's a more long-term, slower response as well. So endocrine gland releasing the hormone, it's going to travel through the bloodstream, it's going to eventually reach a target cell. A target is any body cell that has a receptor for that particular hormone. And so it could be a variety of cells. So for example, estrogen is a hormone. We have estrogen receptors in our ovaries and our uterus and our brains and our bones. I think there's a whole lot more that I'm not thinking of off the top of my head, but my point is there's a variety of different cell types that have receptors for these hormones. So they can kind of cause changes in multiple cell types and stuff at once. Okay, so all of that summarized, nervous system is local, it's rapid, and it's transient or short term. Endocrine is more widespread, slower, and more prolonged response. Some of the kind of broad categories of things that are regulated by the endocrine system: homeostasis, so maintaining our normal composition and blood volume and all of that, maintaining the appropriate volume and blood glucose levels and different ions like sodium and potassium. All of that is regulated by hormones. Controlling the reproductive activity, including development and the function of the reproductive system, expression of sexual behaviors and characteristics, hair growth, changing in body composition, things like that. And then just general development, growth, and metabolism. All of these are kind of broad categories that are regulated by the endocrine system. Some characteristics of endocrine glands. So endocrine cells are derived from epithelia that have secretory function, meaning they have the ability to release molecules. The cells are supported by a framework of connective tissue, and most importantly, there's a large blood supply. That only makes sense because where are our endocrine glands going to release their molecules to? Into the bloodstream. So there has to be a large vascular network or a large blood supply to the endocrine tissue so that they can release those molecules into the bloodstream. So generally speaking, don't get too caught up on the images and diagrams unless I particularly spend a lot of time showing them and walking you through them. I think that some people find the images helpful and some people don't. So, for example, on this slide, I just kind of read these three, keynotes, and didn't really spend any time on this. If looking at this helps you to understand, that's great, but I certainly don't want you spending too much time studying the capillary and the secretory epithelial cells, because unless I've pointed that out to you, and kind of walked you through that, then it's really just there to help you if you need it. So endocrine cells can be organized as one of two different things, either a single organ, so like the parathyroid gland, or just a cluster of cells within an organ that actually serves a different primary function. So we'll see that when we look at the endocrine cells in the pancreas. So it can kind of be its own, organ composed of just endocrine cells, or it can be just clusters of cells within a different organ. Moving on to the parathyroid gland, an endocrine organ. Parathyroid gland are small nodules located on the thyroid gland. So most people have 4, but that number can range from anywhere between 2 and 6 without any major issues. I don't think anybody really knows why there's variation in that. So this large pink butterfly looking or H-shaped thing is your thyroid gland, and you'll notice the nodules on it are your parathyroid glands. Parathyroid glands consist of 2 major types of cells. Chief cells are the ones that release parathyroid hormone. Parathyroid hormone gets released whenever your blood calcium level goes down or becomes low. And the job of parathyroid hormone is to bring those calcium levels back up. So just like we have to keep our blood sugar at our glucose levels in kind of homeostatic range, we have to do that for all of our ions as well, sodium, potassium, calcium. So in this case, if our calcium levels in the blood get low, parathyroid hormone will get released from the parathyroid glands, specifically from the chief cells, and it'll do a couple of things to bring the blood calcium level back up. The other type of cells that you'll find in the parathyroid glands are called oxyphil cells. We really don't know a lot about oxyphil cells. We don't really know what purpose they serve. They are associated with a certain type of cancer known as oxyphil cell adenoma. But in terms of their general role, that's still kind of unknown, and it doesn't seem like they have a role, specifically an endocrine role that we know of. So this slide talks about the structural makeup of hormones. So the hormones are going to fall structurally into one of 3 different categories: steroid hormones, protein hormones, or biogenic amines. So what's going to happen is the hormone is going to be synthesized within an endocrine cell, either starting from a cholesterol molecule, which would lead to the steroid hormone, or from an amino acid, which would turn it into a protein hormone or a biogenic amine. Steroid hormones are lipid soluble, meaning they dissolve well, they travel well in lipid as opposed to aqueous or water-based solution. They're formed from cholesterol, so I don't need you to memorize this exact structure, but you'll notice there's 1, 2, 3, 4 rings on this molecule. So a 4-ringed molecule is what cholesterol is. And then if you change some of these groups on the outside, that might change it from, say, cholesterol to like estrogen or something like that. So if you see this 4-ringed structure and we're talking about hormones, we're talking about a steroid hormone. So these are going to be produced by the gonads, which are going to be the ovaries and the testes, and in the adrenal cortex, which is where we're going to produce cortisol or stress hormone. So when you think of steroid hormones, think of sex hormones and cortisol. And then just know that your sex hormones are estrogen, progesterone, and testosterone. Okay, protein hormone is water soluble. So this one travels well, dissolves well in aqueous or water-based solutions, but not so much in lipid-based solutions. And it's basically just a protein. So if you remember, proteins are nothing more than a long chain of amino acids. So on this picture, these circles or spheres are different amino acids, and they're all chained together to create one protein. In the biogenic amine, these are also water soluble, but there is one exception. Thyroid hormone is the kind of red herring here, because thyroid hormone structurally is a biogenic amine, but in terms of its solubility, it's lipid soluble like the steroid hormones. So biogenic amines are largely water soluble with the exception of thyroid hormone, and they're derived from the amino acid, or they're derived from amino acid that gets kind of modified. So again, this is showing you a modified amino acid here. We're keeping in mind these individual spheres are amino acids, and so this would be just a very small molecule. So what I typically tell people is, if you can remember that your steroid hormones are your sex hormones and your cortisol, and your biogenic amine is epinephrine, norepinephrine, those are your 2 adrenaline molecules, and thyroid hormone then largely everything else falls into the category of protein hormones. So yeah, there are some other steroid hormones and biogenic amines, but for the most part, steroid hormones are going to be sex hormones and cortisol. Biogenic amine is going to be epinephrine, norepinephrine, and thyroid hormone. You could also just remember that as adrenaline and thyroid hormone. And then everything else is going to fall into the protein category. So hormone secretion gets regulated by a reflex. So a reflex is just this pre-programmed response that happens very rapidly when activated by certain stimuli. There's 3 types of stimulation that can cause the release of a hormone. So one of those is hormonal stimulation. This is when a hormone actually stimulates the release of another hormone. The next one is humoral change, where changes in the nutrient or ion composition in the blood cause the release of a hormone. And then the last one is nervous, where you have direct stimulation by the nervous system causing the release of a hormone. So let's look at hormonal regulation. We'll see a lot of this throughout the remainder of this lecture. So hormonal was when a hormone itself actually stimulates the synthesis and release of another hormone. So you have the anterior pituitary, so that is this structure right here. The anterior pituitary releases a hormone called thyroid stimulating hormone, or TSH. That's what these green triangles are. TSH travels through the bloodstream, it reaches the thyroid gland, and stimulates the thyroid gland to release thyroid hormone into the bloodstream. So hormonal regulation, we have the release of one hormone, in this case TSH, stimulating the release of a second hormone, TH, or thyroid hormone. Humoral regulation was when we had changes in nutrient and ion levels in the blood that stimulate a release of hormone. So, classic example of this is our pancreas regulating our blood glucose concentration, or our blood sugar level. So increases in blood glucose levels cause the pancreas to release the hormone insulin. So let's say you just eat a meal, you've digested the meal, it breaks the carbohydrates up into glucose, that glucose goes into your bloodstream, and now your blood sugar has risen. The pancreas has specific endocrine cells that can detect that increase in blood sugar, and they release the hormone insulin. So here comes insulin into the blood, it's going to travel through the bloodstream, and it's going to stimulate the uptake of that glucose by your body cells, which shifts or moves the glucose out of the blood and into the cells, thereby decreasing your blood glucose levels. And then the last one was nervous regulation. So this is when you have direct activation of the nervous system stimulating the release of a hormone. So a good example of this is when the sympathetic nervous system, if you remember, that's your fight or flight, that's your emergency mode. When the sympathetic nervous system increases in activity, so something's kind of stressing our body, the nerves stimulate the adrenal medulla, that's the center region of your adrenal gland, to release the hormones epinephrine and norepinephrine. Remember, those are your adrenaline molecules. So we're in fight or flight mode, there's some kind of emergency, and we need adrenaline in our body. So here's part of our spinal cord. You have this neuron that's going to come, and the neuron is going to directly stimulate the adrenal medulla, which is where that norepinephrine and epinephrine are going to be released from. So we have nervous system stimulation causing the release of our adrenaline hormones. Hormones, by definition, are going to be transported within the bloodstream. That's basically what they are. They're molecules released by endocrine cells that travel through the bloodstream. For those water-soluble hormones, which if you remember are the protein hormones and the biogenic amines, they simply dissolve in the blood plasma and they travel throughout the bloodstream with no major issues to get to their target cell. But the lipid-soluble hormones, so the steroid hormones such as calcitriol, thyroid hormones, estrogen, progesterone, testosterone, all of those, they require what are called carrier molecules, because remember, those are lipid-soluble. They don't travel well or dissolve well in aqueous solutions, which our plasma is. So they have a carrier molecule. Think of it like a shuttle molecule, and that assists them with being transported throughout the blood. So what a carrier molecule is it's a water-soluble protein that's been produced by the liver. So it goes out into the bloodstream. It can grab a hold of that lipid-soluble hormone and basically just kind of shelter it and help it move throughout the bloodstream. So albumin is a carrier protein. It's kind of a generic one. It can carry a bunch of different lipid-soluble molecules or hormones. But then we also have what are called binding globulins, which are proteins that are specific for a particular hormone. So thyroxine binding globulin is specific for thyroid hormone. Then you have sex hormone binding globulin, which is specific for your sex hormones. So if we're talking about these lipid-soluble hormones, and then we know that they're being transported through the blood attached to a carrier molecule, then we need to discuss bound versus free, because it's only the unbound or the free hormone that's actually available to leave the blood and bind to its receptor on the target cell or the target tissue. If it's bound up by a carrier molecule, then it's kind of tied up and it can't work. So you've got total hormone, which is the total amount of hormone circulating through your blood, which includes both the free and the unbound. I'm sorry, both the free and the bound. Because while I mentioned that the lipid-soluble hormone does have to travel through the carrier molecule, there's always some percentage of it that is not attached to a carrier molecule. Very small percentage, but there is some that is free. So the free hormone makes up only about 0.1 to maximum 10% of the total hormone circulating through the blood. The remainder of that's going to be bound. This particular picture, we're going to go through actually this in detail in a minute, but for this particular one, I don't need you to really spend a lot of time looking at this. What I really wanted to show you is this says SHBG, that stands for sex hormone binding globulin, so this is the carrier molecule. This is testosterone. This is the plasma membrane, so everything inside here is the cell, everything outside here is the blood. So the hormone is traveling attached to the carrier molecule through the blood. At some point it will detach and become free, and it's only this free hormone that is then available to move inside the cell and do all the things it's going to do. So what does a unbound hormone do? So again, it's kind of the same thing. The blue is our carrier molecule. The orange is our steroid hormone. Here's our bloodstream. Here's our cell with the plasma membrane being right here. And this would be the cytoplasm, nuclear membrane, and inside the nucleus you can see the DNA. So there's your orientation. Okay, so right here the carrier protein with the hormone is just traveling through the blood, and you can see at some point it kind of pops off, diffuses off of the carrier protein. So now it's free. So free hormone is free to do its job. So this is a lipid soluble, right? And if you are asked the question, how do I know this is lipid soluble? Because only the lipid soluble molecules travel attached to a carrier protein in the blood. Okay, so we know this is lipid soluble. Do we remember what the plasma membrane is made up of? Phospholipid bilayer. This is a lipid layer. So because this is a lipid soluble hormone and this is a lipid layer, it very easily diffuses across. Normally the plasma membrane is kind of like a barrier. It's like a gateway to like keep things out and allow certain things in when it wants to, but when it's a lipid-soluble molecule, it can diffuse right across. Okay, so our hormone detached from the carrier molecule, it diffused out of the bloodstream across the lipid bilayer, and it found its receptor on the inside of the cell. So here's the receptor for this hormone. The hormone will bind to that receptor, and it forms what's called a hormone receptor complex. The hormone receptor complex then moves into the nucleus. So we're moving into the nucleus. Here it is right here. And it binds to a very specific sequence of DNA called a hormone response element. So here's your DNA. It's this long chain of nucleotides. And you'll have a pattern of those nucleotides that are specific to binding to this hormone receptor complex. And that specific pattern is the hormone response element. So, simple area of DNA called the hormone response element that the hormone receptor complex binds to. So, here it is sitting down there. And that is going to initiate the process of transcription. So that goes way back to pretty early on in 2401. Transcription is where we transcribe an mRNA molecule from our DNA for a particular gene. And then that mRNA will leave the nucleus, come out into the cytoplasm, hook up with a ribosome, and undergo the process of translation, also from 2401. So if you need to review those, you might go back and review those. But translation is when you translate that mRNA transcript into a protein molecule. So what we started with was a hormone bound to its carrier protein in the blood, and we ended with the synthesis of a new protein. So this kind of shows you the full picture of how a hormone molecule leads to some kind of metabolic or biochemical change within the cell. So I'll run through that again. I do want you do need to know all of these kind of steps and processes, and know that these specific ones are just associated with your lipid-soluble molecule or lipid-soluble hormone. So it's traveling through the blood, attached to the carrier protein. At some point it detaches. The hormone crosses the plasma membrane, goes into the cytoplasm. It binds to its receptor, so forming the hormone receptor complex. That entire complex then enters into the nucleus. It finds the appropriate hormone response element, which is that pattern on the DNA. So for example, if this is estrogen, it's specifically called an estrogen response element. It sits down on the DNA. It initiates the process of transcription. So we end up with an mRNA molecule, which leaves the nucleus, goes into the cytoplasm, hooks up with a ribosome, and starts the process of translation, which leads us with a new protein. So the lipid-soluble hormones were able to cross the plasma membrane because they're lipophilic, meaning they like the lipid environment, and the plasma membrane is just that. It's made up of phospholipids and cholesterol, which is also lipid, so it's a very lipid-rich environment. So because they can easily cross in, then they get to their receptors, which are located within the cytosol of the cell. Now, look at the water-soluble hormones. They like aqueous environments, or water-based environments. The blood or the plasma is aqueous. The inside of the cell is aqueous as well, but that plasma membrane is not. So the water-soluble hormones are not going to be able to penetrate or simply go through that plasma membrane layer. So water-soluble hormones are what we refer to as hydrophilic because they like the water as opposed to the lipids. So how do they bind to their receptors? Well, basically, we have to change where their receptors are located. So their receptors are not located in the cytosol, but rather within the plasma membrane. And they have an exposure of part of their receptor on the outside of the plasma membrane that would make contact with the blood. And so that's how the water-soluble hormone can activate its receptor. So I should have had a title on the slide, and I want you to make a note of this particular slide, that this one and the next couple slides are explaining what happens when a water-soluble hormone makes it to its cell, to its receptor on the cell. So we talked about what happens with the lipid-soluble hormone, and it eventually leads to transcription and translation. So here we're talking about what happens when the water-soluble hormone gets there. So you can see the exposure to the outside of the, this is not going to work. So we're going to talk about two different second messenger pathways. So this slide is picking up where the last one left off. We have this activated G protein with an attached GTP molecule, and it's traveling on the underside of the plasma membrane. And so this is called the adenylate cyclase pathway. So here's an adenylate cyclase molecule. So the activated G protein is going to bind to this enzyme called adenylate cyclase. That's going to activate the enzyme. Adenylate cyclase is responsible for converting ATP into a molecule known as cyclic AMP. And then cyclic AMP goes on to activate a molecule called protein kinase A. Protein kinase A is, as its name suggests, a kinase. And I don't know if you specifically discussed kinases in 2401, but a kinase is a protein that activates other intracellular proteins. So every once in a while it will do the opposite and inactivate it, but say 99% of the time a kinase activates other molecules in the cell. And it does that by adding a phosphate. It phosphorylates them, so it adds a phosphate group to them. And when you add a phosphate group to something, think of it as turning the light switch on. You put phosphate on, it turns it on. It's going to do its job. When it's done doing its job, you take the phosphate off. That's called dephosphorylation. That's not done by kinases. Kinases only add the phosphate. But when you take that phosphate off, it's like turning the light switch off. You deactivate it. Okay, so in the previous slide, we generated this activated G protein and it detached from the hormone receptor and it's traveling. In this pathway, it attaches to adenylate cyclase, activating that enzyme. That enzyme causes the conversion of ATP into cyclic AMP. Cyclic AMP goes on to activate protein kinase A, and then protein kinase A will then continue on and activate different proteins and enzymes in the cell. So let's contrast this to what we saw with lipid-soluble, where the lipid-soluble hormone led to the creation of new proteins within our cell. Remember, we had the transcription and the translation. In this example, we're not making new proteins, we're actually turning on proteins that were already there. So it's a more efficient way of changing the activities in their cell. We don't have to go through the whole process of transcription and translation. The proteins are already there, but they're turned off. And when the hormone comes and we activate our G protein and we create second messengers, those second messengers, in this case cyclic AMP and protein kinase A, just turn on the proteins and the enzymes that are already there. So they just start working and lead to biochemical changes inside the cell. This is an alternative second messenger pathway. So this is a phospholipase C pathway. So there's two second messenger pathways, adenylate cyclase on the previous slide, and then phospholipase C on this slide. Okay, so now we're picking up where the activated water-soluble hormone receptor had caused activated G protein. And in this pathway, instead of running into adenylate cyclase, it's running into a molecule known as phospholipase C. So it's going to attach itself to phospholipase C. Phospholipase C always sits in the membrane next to a lipid molecule known as PIP2. Whenever the G protein activates phospholipase C, it cleaves this PIP2 into two different molecules. One of them is IP3, and one of them is diacylglycerol, or DAG. If you just know it as DAG, that's fine. So this PIP2 splits into IP3 and DAG. IP3 goes on to the endoplasmic reticulum, and it causes the release of calcium. And then calcium comes out into the cytosol, and it can do a number of different things. I won't go into all the things that calcium can do, but calcium itself is a second messenger. DAG goes on and activates protein kinase C. And protein kinase C acts just like protein kinase A did in the other pathway. It's a kinase. It will go on to phosphorylate other intracellular proteins, thereby activating them, turning them on so that they can do their job. So, activated G protein binds to phospholipase C. That causes the splitting of PIP2 into IP3 and DAG. IP3 causes the release of calcium stores from the endoplasmic reticulum into the cytosol. So calcium will go on to do a number of different intracellular activities. DAG activates protein kinase C, which will then phosphorylate and activate other proteins in the cell. So let's look at specific examples of how the second messenger pathways work. Okay, so we're going to look at glucagon for now. Glucagon is released from pancreatic cells in response to low blood glucose level. So if you're fasting or you're starving and we haven't had any dietary glucose taken in, our blood sugar is going to drop. Our pancreas detects that and releases glucagon. Okay. Glucagon is going to go into the bloodstream. It's going to travel to the liver, and it's going to combine with the receptor that's located in the plasma membrane of the liver cells, which is going to lead to an increase in cyclic AMP synthesis and an activation of protein kinase A enzymes. So this was that adenylate cyclase pathway. Remember, it activated cyclic AMP, activated protein kinase A. Protein kinase A enzymes will phosphorylate specific enzyme pathway that lead to the release of glucose from your liver cells. And so we're going to start releasing glucose stores from our liver cells into our bloodstream, and that's going to increase our blood glucose levels and get it back to normal. So we had a physiological change. We had a drop in blood glucose level that was detected by our endocrine cells, which released glucagon. Glucagon traveled through the bloodstream. It's a water-soluble hormone. It bound to its receptor on the liver cells. That was adenylate cyclase pathway. So we activated a G protein, adenylate cyclase, cyclic AMP, and protein kinase A from that one slide. Protein kinase A phosphorylates and activates enzymes that are required to release glucose from those liver cells. So when that happens, glucose leaves the liver cells, goes into the bloodstream, and it brings our blood glucose level back up to normal range. So oxytocin is an example of the other pathway, the phospholipase C pathway. So oxytocin is a hormone that gets released from the posterior pituitary during active labor, during childbirth. It's going to travel through the bloodstream down to the uterine muscle. It's going to bind with its membrane receptors of the smooth muscle cells in the uterus and activate that G protein and activate the phospholipase C. And then remember, phospholipase C causes the splitting of PIP2 into DAG and IP3. Now in this case, IP3 is going to go down to the sarcoplasmic reticulum, increase intracellular calcium levels, and if you remember from 2401, that calcium is very important for allowing muscle contractions. So upon the release of the intracellular stores of calcium, where it's going to cause stronger uterine contractions, and that's going to help expel the baby. So how our cell and how our body responds to a certain hormone is highly influenced by the number of receptors that are being expressed for that hormone, and that the number of receptors can change. It's very dynamic. It's going to change due to a variety of factors, but we can have a lot of hormone receptors expressed on the cell surface, or we can have very few. So a lot of times that depends on what the level of hormone is that's currently circulating. So imagine that we have very low circulating levels of hormone. Then we're not going to have a very robust response because we just don't have a lot of hormones circulating. So what we want to do in that case is increase the level of receptors that are expressed on our cell surface. It's kind of like if we don't have a lot of hormone going around, we don't want to miss one. We want to be sure we get every single molecule of hormone that passes by. So let's put out more receptors so we catch them all. And that's called upregulation. So upregulation is when you increase the number of receptors on your cell surface, typically in response to low circulating hormone levels. Contrary to that, if you have really high circulating levels of hormone, then you're going to get a really strong, robust signal, and sometimes it's more than you want. So what you can do is pull some of those receptors into the cell, which if they're completely in the cell, they cannot bind the hormone. And that's called downregulation. So you're going to decrease the number of receptors that are exposed on the cell surface, and that kind of turns down the signal, so to speak. So this is a really silly analogy, but it's how I kind of envision upregulation and downregulation. So on my head, I have 2 ears, and I can hear you. If you start speaking really quietly and my 2 ears are not enough, if I can pull another 4, 5, 6 ears out of my pocket and attach them to my head, I'm more likely going to hear you better. If that would be upregulation. I'm pulling more ears out to attach to my head so I can increase my sensitivity so I can hear your whisper. Okay, so now I go back to my 2 ears, and if you start shouting and screaming and you're talking so loudly, I'm going to start pulling ears off, reducing the amount of ears so that I don't get so much of that signal. So it kind of tunes it down. So totally ridiculous example, but that's basically what upregulation and downregulation is. We're changing the numbers of receptors that are expressed on our cells to kind of accommodate or compensate for the circulating hormone levels so that we maintain the appropriate signal. This also has to do with developing a drug tolerance. So if we're taking a medication, a lot of times it's something that our body wouldn't have normally, but we just need it for the time to treat some kind of situation. But our body's constantly trying to maintain this kind of homeostasis balance. So when you take this, let's say we're talking about, opioids. Let's say you're taking opioids for something, and it's not something that your body normally sees a lot of. So you take a certain dose and you feel a certain way, and it's usually a good way. And then, but your body says, well, we're not supposed to have that level of response. So it starts to downregulate the receptors. So now you have less receptors on your cells. So then the next time you go to take a dose, or, a week later and you take more, you take the same dose, but you didn't get the same response because you downregulated your receptors. That's your body's natural way of trying to tone down the signal that you gave it. Now what happens here is that some people, they take that next dose, they don't get the same response, so what do? They take more. So now they're at a higher dose, but they're still not getting the response, and your body continues to downregulate, and in response that person continues to take more and more, and that's how you can develop this kind of tolerance to a drug where you're now requiring higher and higher doses and not getting the same effect. So hormones interact with one another in a variety of ways. So a cell's response is going to depend on the interaction of the pathways induced by whichever hormones are around that are all acting on that one cell. So you can have a synergistic response where hormones work together to produce a greater effect. So you see that in the female reproductive tract. Estrogen and progesterone work together to produce greater effects than just one or the other. You have permissive relationships where one hormone allows the action of the second hormone. So we see this during lactation, where prolactin is the hormone that produces the breast milk, but then oxytocin is the hormone that allows milk ejection. So you have to have the prolactin first to make the milk so that oxytocin can do its job for milk ejection. So that's permissive. One has to happen first to allow the second one. Then you have antagonistic effects, where one hormone causes the opposite effect of the other. Classic example of this is insulin and glucagon. Now, these two hormones are not acting together. We will either have an increase in glucagon, or we will have an increase in insulin, but we do not have the two of those hormones fighting each other at the same time because they have two very different stimulation. Insulin gets released when your blood sugar goes up. Glucagon gets released when your blood sugar goes down. Nevertheless, they have opposite effects. So insulin induces changes that lead to a decrease in blood sugar, whereas glucagon induces changes that lead to an increase in blood sugar. Okay, so this slide is in here kind of as a reference slide. I'm not going to spend much time on it, and I don't need you to go through and memorize this, but in some of our later slides, we're going to use some of this terminology, and the words are very similar, but they mean very different things, so I wanted you to have this to go back to. So glycogenesis is the formation of glycogen from glucose. Glycogen, you remember, it's just our kind of glucose storage molecule. It's just a long chain of glucose that when we have extra glucose, we create glycogen, we store it in the liver and the skeletal muscles. So glycogenesis is the formation of the glycogen. Glycogenolysis is the breakdown of glycogen. So when we have too much glucose, we're going to form glycogen through glycogenesis. When we have a lack of glucose, we're going to break it down through glycogenolysis. Gluconeogenesis is the formation of glucose from non-carbohydrate sources. And so this was all glucose related. And then when it comes to our fatty acids, you have lipogenesis, formation of triglycerides from glycerol and fatty acids. Lipolysis is the breakdown of those triglycerides. And then protein anabolism is the buildup of protein. Protein catabolism is the breakdown. So again, it's just a reference slide because when we get to some of those later slides and you'll see some of these terminologies, I didn't want you to get lost. I want you to have this to go back to. Alright, so let's get started on the hypothalamus and pituitary gland. So the hypothalamus is known as the master endocrine gland because it regulates the hormonal secretion of other endocrine glands, one of those being the pituitary gland. So let's get our bearings. Here's the brain. We're looking right in this region. Okay, so zoom out. This is our hypothalamus right here. These are just showing you different bodies of cells that, kind of reside in the hypothalamus. Lying inferior to the hypothalamus is the pituitary gland. So all of this is the pituitary gland, and they're connected by what's called the infundibulum. So this little stalk right here connects the hypothalamus to the pituitary gland. The pituitary gland lies in that cella turcica of the sphenoid bone. If you remember that from 2401, the sphenoid bone was that butterfly-shaped bone, and it had a little indentation, which allows the pituitary gland to kind of sit right in there. Now the pituitary gland is divided into anterior and posterior sections. So let's look first at the interactions between the hypothalamus and the posterior pituitary. So again, here's the hypothalamus, here's the infundibulum, and this would be the posterior pituitary. So the posterior pituitary only releases two hormones, oxytocin and antidiuretic hormone, or ADH. So you have these neurons, and they have their cell body in the hypothalamus, and they have these really long axons that travel down through the stalk, and their axon terminals lie within the posterior pituitary. So up here in these cell bodies, this is where we're going to synthesize ADH and oxytocin. And then we're going to make the hormones, and we're going to travel down to the axon terminals, and then they're going to wait here. So whenever there's a stimulus that requires or stimulates the release of one of these hormones, you'll get an action potential, and then that will cause the release of either ADH or oxytocin into the systemic circulation. Now it looks a lot different and a little bit more complicated when we talk about the interactions between the hypothalamus and the anterior pituitary. So the anterior pituitary releases a whole bunch more hormones. Thyroid stimulating hormone, or TSH, follicle stimulating hormone, or FSH, luteinizing hormone, or LH, adrenocorticotropic hormone, or ACTH, growth hormone, GH, and prolactin, which is PRL. All of those come from the anterior pituitary. Now, one of the things you'll notice here is here's your hypothalamic neurons, so the neurons that have the cell body in the hypothalamus. They are not coming all the way through the stalk and reaching down to the anterior pituitary like they did in the posterior pituitary. Rather, they're reaching all the way down right here, and you'll notice this little capillary bed. This is called the primary capillary plexus. So what you have is releasing hormones, and sometimes inhibitory hormones, that are made in the hypothalamus. And they go down, and we'll use growth hormone for example. We have growth hormone releasing hormone that gets made by the hypothalamic neurons. That gets released into the primary capillary plexus. Growth hormone releasing hormone then travels down through this blood system. This is called the hypophyseal portal system, and it stimulates the cells of the anterior pituitary to release growth hormone. So then it comes out, and the growth hormone goes into the systemic circulation. Okay, so the neurons don't reach all the way down to the anterior pituitary. They only reach down to the primary capillary plexus. So whenever they're stimulated, the hypothalamic neurons will release either releasing hormones or inhibitory hormones into this primary capillary plexus. Those hypothalamic releasing or inhibitory hormones travel down through the portal veins to the anterior pituitary and stimulate the cells here to either stimulate or inhibit the release of the hormones here from the anterior pituitary. So if it's a stimulation, then the anterior pituitary will release the appropriate hormone. It would be one of these: TSH, FSH, LH, ACTH, GH, or prolactin. And then from there, it gets released into the systemic circulation. So again, these are some of the hormones that come from the anterior pituitary, kind of telling you what they are. Thyroid stimulating hormone goes down, stimulates the thyroid to release thyroid hormone. So this really shows you the complexity of this. Let's look at just thyroid hormone. In order to get thyroid hormone, you have the hypothalamus releasing thyrotropin releasing hormone. That's the hormone that goes into that primary capillary plexus. That stimulates the anterior pituitary to release thyroid stimulating hormone. That goes into the systemic circulation and stimulates the thyroid to release thyroid hormone. So it's very complex. There's a lot of steps, but it really allows our body to have a very fine-tuned regulation of our hormones. Prolactin acts on the mammary glands to stimulate milk production. FSH and LH, these are follicle stimulating hormone and luteinizing hormone. We're going to actually talk about these when we get to the reproductive system, so we're just kind of ignoring those for now. Growth hormone acts on just about all your body cells and tissue to promote growth. And then adrenocorticotropic hormone acts on the adrenal cortex to release corticosteroids, or your stress hormones. So again, I put this in here just to show you, one, that the posterior pituitary releases oxytocin and ADH, and this tells you where they work, and that the anterior pituitary releases these molecules. So thyroid stimulating hormone goes to the thyroid, adrenocorticotropic hormone goes to the cortex, FSH and LH goes to the gonads. We'll talk about those in reproductive. Growth hormone pretty much goes everywhere. Prolactin goes to the mammary glands, and then endorphins we're not really covering, so you can also kind of ignore those. But I also wanted to put this in there so that you can see, because you can't always say anterior pituitary is on the right, posterior is on the left, or vice versa. So how can you, from a very general diagram, determine what's anterior and posterior? You can look at the neurons from the hypothalamus. In this case, the hypothalamic neurons are reaching all the way down to the pituitary, and that only happens in the posterior pituitary. Okay? In the anterior pituitary, remember those hypothalamic neurons only reach down to the primary capillary plexus. So you can look at those neurons, and that can tell you if you're looking at the anterior or the posterior pituitary. So this slide is looking specifically at growth hormone. So let's start over here with how we get the release of growth hormone. I kind of went through it in one of the other slides. So you have neurons from the hypothalamus, and they're going to release growth hormone releasing hormone. They release that into the primary capillary plexus, and the growth hormone releasing hormone travels down to the blood vessels located within the anterior pituitary, and that stimulates the cells of the anterior pituitary to release growth hormone. So that growth hormone then goes out into the systemic circulation, and these down here show you some of the effects that it has. So glucose sparing effect, so it's going to stimulate the adipose cells or the fat cells to break down your stored fat, and the effect of that is going to be that it's going to promote growth effects. You'll also see an increase in the uptake of amino acids from the blood and enhanced cellular proliferation. That means cellular reproduction, so we're making more cells, and reducing apoptosis. So again, apoptosis is when cell dies. So we're kind of promoting cell growth and development and reproduction and reducing cell death. And this kind of shows you some of the main cells that are affected by growth hormone. It also has what's called a diabetogenic effect, meaning that the growth hormone will stimulate the liver to break down glycogen. Remember, glycogen were those long chains of glucose stringed together, stored primarily in our liver and our skeletal muscle. So we're going to break down glycogen, and when we do that, it releases individual glucose molecules into the blood. So putting glucose into the blood is ultimately going to promote growth effects. One of the other effects of the liver is that you're going to get the release of something called IGF-1. So IGF-1 starts to come out. IGF-1 further stimulates the growth effects that are already happening. But IGF-1, importantly, also has a negative feedback role. So as we continue to release IGF-1, the levels get higher and higher, and that feeds back to the hypothalamus. And whenever the hypothalamus detects higher levels of IGF-1, that's basically a signal that we have enough growth hormone and we don't need to continue to release more. And so what it does is it starts to release growth hormone inhibitory hormone. And so growth hormone inhibitory hormone then comes down, goes into the primary capillary plexus, and that will travel down to the anterior pituitary to slow down or prevent the release of any more growth hormone. So it all started with growth hormone releasing hormone, getting released into the primary capillary plexus, that travels down and stimulates the release of growth hormone. Growth hormone has all of these different effects. It breaks down fat, takes up amino acids, it promotes cellular proliferation and reduces apoptosis, and then it stimulates the liver to break down glycogen, which promotes higher blood glucose level, releasing glucose into the blood. Also releases IGF-1. IGF-1 essentially enhances all of these other effects, but also IGF-1 feeds back to the hypothalamus, serves as an indicator that we have sufficient levels of growth hormone so that the hypothalamus now releases growth hormone inhibitory hormone, which then travels down to the primary capillary plexus and essentially tells the anterior pituitary to stop the synthesis and the release of growth hormone. Let's look at some of the disorders that are associated with abnormal growth hormone secretion. So one of those is pituitary dwarfism. So pituitary dwarfism exists at birth, but it's not really obvious until about 1 year old in the child. It's caused by inadequate growth hormone production, which can be due to some problem in either the hypothalamus or the pituitary gland. You know that growth hormone itself is released from the anterior pituitary, but growth hormone releasing hormone is released from the hypothalamus. So it could be a dysfunction in either of those two areas that results in inadequate secretion of growth hormone that can lead to pituitary dwarfism. So growth hormone injections can help by replacing some of the growth hormone that's not being released, but it really has kind of a limited extent. It can't fully restore growth. On the opposite end of things, you have something called gigantism, which is caused by excessive growth hormone production in children, specifically in children. So it's going to cause extraordinary heights, upwards of 8 feet tall, high blood glucose levels, large internal organs, and a large tongue. Gigantism usually causes death at a relatively early age, primarily due to heart failure or complications with blood sugar. Now, another disorder that has to do with excessive growth hormone production is called acromegaly. The distinction between acromegaly and gigantism is that acromegaly is caused by excessive growth hormone production, specifically in adults. Gigantism is excessive growth hormone production, specifically in children. So let's say somebody has normal growth hormone release as a child, but after puberty as an adult, they start, for whatever reason, to have excessive growth hormone production. That would then lead to acromegaly. So it doesn't affect the height because this individual is post-puberty, the growth plates are fused and they're essentially done growing. So height is not effective, but they do have continued bone growth in certain areas, such as their hands, their feet, and their facial bones. And you can see this individual with acromegaly with fairly normal proportions as a young child, a teenager, but clearly as she continues to age, the hands and the facial bones, you can tell, are enlarged. So it also will lead to increases in internal organ size. The increase in the liver size results in increased glucose release, and it can lead to diabetes. And oftentimes this is caused by a loss of a negative feedback by a pituitary tumor. So if you need to refresh yourself on negative feedback and positive feedback systems, this would be a good time to do that. Essentially, when there is a need for a molecule in our body will produce it. And then once we have sufficient amounts of that molecule in our body, it feeds back to the brain and tells us we have enough. So if we're not getting that feedback, then our brain does not know to then shut it off, and it will continue to release and release and release, and that's going to lead to excessive amounts. And so that's one of the things that you could have here with acromegaly. Moving on to the thyroid gland. The thyroid gland lies just anterior to the trachea, so you could see the trachea here, and this H or butterfly shaped thing right here is your thyroid gland. It's composed of a right and a left lobe here that kind of join here in the center. So this slide kind of walks you through how we synthesize and release thyroid hormone. So our thyroid gland is made up of what are called follicular cells. And follicular cells kind of make this surrounding, so here's a follicle. So it's made up of follicles. The outside is made up of follicular cells, and the inside is this kind of gelatinous substance called colloid. So what you're looking at here is one follicle, these being the follicular cells. This inside here is the colloid, and then of course you have your blood vessel passing on top of it. So this just goes kind of step by step to explain how it's synthesized and released. So iodide gets transported out of the blood and into the follicular cells, where two of them join together to form I2. Now, meanwhile, at the same time that's going on, the follicular cell is doing some protein synthesis to synthesize a protein called thyroglobulin. So this is thyroglobulin. You'll see that it has tyrosine here, just pointing out certain residues because there's tyrosine is a specific amino acid, and there's a few of them in the thyroglobulin molecule. Okay, so we have I2 coming in here. We have the synthesis of thyroglobulin, and then both of those leave the follicular cell and go into that colloid area, that center region of the follicle. So all that's coming down into the colloid. Now, one I2 molecule can bind to the tyrosine on the thyroglobulin molecule. So see how this was a thyroglobulin, and then you have a couple of tyrosines. So we're zooming in, so here's that tyrosine molecule. You can have one I2 bind onto it, and it's called monoiodotyrosine, or MIT, because it's just got one. Alternatively, you could get two I2 molecules that would bind to the tyrosine, and that would lead to diiodotyrosine, or DIT. So the I2 is going to hook on to the tyrosine amino acid that's part of that larger thyroglobulin protein molecule, and if you get one attached, it's called MIT. If you get two attached, it's called DIT. Now the next thing that's going to happen is we're going to get some combining of our MITs and DITs. So if we have an MIT connect to a DIT, count the iodines. You have 1, 2, 3. That's called T3. It says pre-T3 right here because ultimately we're going to do some other modifications to it, but it's going to essentially wind up as T3. If we have a DIT connect to another DIT, you have 1, 2, 3, 4 I2s on there, so you get T4. T3 and T4 are your thyroid hormones. So again, it says pre-T3 and pre-T4 because at this point they're going to go back into the follicular cell. They're going to undergo some modifications, and ultimately they're going to then be released from the follicular cell into the bloodstream as mature T3 and T4. So from there, they're in the bloodstream. That's your thyroid hormone, and they can go on to do their job. So looking at the overall hypothalamic pituitary thyroid axis, or HPT axis, so that refers to the communication and interaction between the hypothalamus, the pituitary, and the thyroid gland. So I find these diagrams very helpful, so I would study these and understand the different stimuli and the different steps that go into this whole process. So first thing that has to happen is there's a stimulus. So the hypothalamus could be stimulated by maybe the fact that there's decreasing or low thyroid hormone that's circulating, and then here's some other ones: cold weather, pregnancy, high altitude, hypoglycemia. All of those can serve as a stimulus to the hypothalamus, telling it essentially we need to start the process of making more thyroid hormone. Okay, so we have stimulation, so the hypothalamus then responds to that stimulation and it releases thyrotropin releasing hormone. So at this point I'm over here in the step. Thyrotropin releasing hormone, or TRH, goes into the hypothalamal hypophyseal portal system. It's releasing that into the primary capillary plexus, and that sends it down then to the anterior pituitary to stimulate the release of thyroid stimulating hormone, or TSH. So here you have TSH leaving the anterior pituitary. Here it is in the steps. And it's going to travel through the systemic circulation until it reaches the thyroid gland. When the TSH reaches the thyroid gland, it stimulates the process that we just showed on the previous slide, the synthesis and the release of thyroid hormone. So here comes thyroid hormone out of the thyroid gland. It's going to go into the systemic circulation, and then the blue box down here tells you all of the things that are going to happen as a result of that thyroid hormone being released. So you're going to have an increase in uptake of amino acids. That's going to happen in virtually all of your cells, but especially in neurons, and that will result in an increased in metabolic rate and an increased glucose uptake. You're going to have increased glycogenolysis and gluconeogenesis. So this is where that one slide that I said earlier was more of a reference slide. If you're confused by this terminology, you can go back to that to see what it means. Increased glycogenolysis means we're going to break down our glycogen stores and release individual glucose molecules. Gluconeogenesis means we're going to start to create glucose from non-carbohydrate sources. So ultimately the result of this is going to be the liver will be releasing glucose into the bloodstream. And then decreased glycogenesis. So glycogenesis is when we create glycogen. Well, we certainly don't want to create glycogen if we're currently in the process of breaking it down. That would be a waste of energy. So we're not going to be doing glycogenesis. Okay, so amino acids, we had an uptake in our cells for amino acids. We've got the liver releasing glucose. We're going to have an increase in lipolysis, meaning we're going to start to break down our triglycerides into glycerol and fatty acids, and that's going to move out into the bloodstream. And then on a larger scale, you're going to get an increase in breathing rate and an increase in heart rate and force of contraction. So ultimately, we're increasing our metabolism rate by thyroid hormone. So we've put out all the molecules we need to. We've got glucose coming into the blood. We've got fatty acid coming into the blood. Those are energy molecules that our body can use to fuel this increased metabolism. Well, by increasing our breathing rate and increasing our heart rate and force of contraction, that's basically going to provide the oxygen that is going to also support that increased metabolic demand from thyroid hormone. So we've got our stimulus. It's showing you stepwise where we're starting, the pathway that we're taking, the different hormones that are coming out of the different tissues, and then again down here in the blue box are all of the effects that that final release of thyroid hormone is going to have. So thyroid hormone is a lipid-soluble hormone. Remember, it belongs to the category of biogenic amines, which most biogenic amines are water-soluble, but thyroid hormone is the exception. It's a lipid-soluble hormone, and what we know about that then is that it has to travel through the blood bound to carrier proteins. So albumin was the carrier protein. That's kind of a general carrier protein. It can carry a wide variety of different hormones, but then there's also thyroxine binding globulin that binds specifically to thyroid hormone. Now, we talked about how thyroid hormone, we have T3 and T4. T3 is the most active form of the hormone, but T4 is the most abundant. So, how do we get about that is most of our cells have an enzyme that converts T4 into T3, and basically what it's doing is just removing an iodine molecule. And so that's how we can create more T3 since it's the more active form. We just remove an iodine from T4, and that essentially makes it T3, and then it's more active. So talking about some of the disorders of thyroid hormone secretion, Graves' disease is one of them. Graves' disease is a form of hyperthyroidism, meaning too much thyroid hormone. Characteristics of this are increased metabolic rate, weight loss, hyperactivity, heat intolerance, and exothalamus, which is where you have these protruding or bulging eyeballs. This can be caused by an irregular immune response where an antibody is generated that acts like thyroid stimulating hormone. So if you can think about our feedback mechanism, we had thyrotropin releasing hormone coming from the hypothalamus, stimulates the release of thyroid stimulating hormone, which then stimulates the release of thyroid hormone. So normally when you have enough thyroid hormone, it's going to feed back to the hypothalamus. We're going to stop releasing the thyrotropin releasing hormone, or the TRH, and that's going to stop releasing the TSH. But imagine that you have an antibody that looks and acts like thyroid stimulating hormone. Because it's not actually TSH, it's not part of that feedback system. So even if the feedback system is working correctly, you have this other molecule unaffected by that looks and acts like TSH. So what's going to happen is it's going to continue to stimulate the thyroid gland, and then the thyroid gland is going to continue to release thyroid hormone. It will be unaffected by what would normally be your normal negative feedback mechanism. So this can be treated by surgical removal of the thyroid gland. We can just take it out, and so that we don't have excess, but since we still need thyroid hormone, then we're going to have to supplement with daily thyroid hormone supplements. Another disorder of thyroid hormone secretion is hypothyroidism. So this is where we have insufficient levels or low levels of thyroid hormone. Characteristics here are going to include low metabolic rate, lethargy, which is kind of a feeling of tiredness, a feeling of being cold, weight gain, and photophobia, which is the avoidance of light. So this can be caused by insufficient dietary iodine. We saw when we looked at the synthesis of thyroid hormone what an important role iodine plays in that. It can also be caused by loss of stimulation by the pituitary gland. So if the pituitary gland isn't kind of getting the signal from the hypothalamus that it needs to release TSH, then it doesn't release it, and therefore a thyroid gland doesn't release thyroid hormone. Or it can also result in an abnormal immune response that results in the destruction of the thyroid gland. So this is autoimmune. So autoimmune is where our immune system, as opposed to its normal role of attacking foreign pathogens or bad things such as bacteria and viruses, something kind of goes wrong in the system and it can start to attack our normal natural body cells. So if we have an autoimmune response attacking the thyroid, then that prevents it from being able to do its job and release thyroid hormone. So regardless of the cause, if there is insufficient thyroid hormone being released, we can treat that pharmacologically using thyroid hormone supplementation. Now another one is called goiter. Goiter is where you have an enlargement of the thyroid gland due to insufficient dietary iodine, so you can see that over here. So what happens in this case is your pituitary is going to release more and more TSH to stimulate the thyroid. So we don't have enough thyroid hormone, so it's going to tell our hypothalamus that. The hypothalamus is going to release TRH. TRH is going to tell the anterior pituitary to release TSH. And TSH goes down to the thyroid and tells it to make thyroid hormone. But if there is no iodine, then we cannot make thyroid hormone. So we never end up releasing enough T3 and T4. So the pituitary continues to release more and more and more and more TSH, really kind of yelling at the thyroid gland saying, we need you to make your thyroid hormone. Well, eventually that overstimulation causes the thyroid follicles to kind of overgrow and enlarge, and that's what results in this enlarged thyroid gland. So this used to be common in the United States until iodine was added to the table salt. So now dietary iodine is pretty much a given. It's provided in our table salt. But it does still exist in areas of the world where there's low dietary iodine. So if you start to replace the dietary iodine, it will not cause the goiter to regress. So surgical removal is often required to get rid of it. Moving on to the adrenal glands, we have two adrenal glands. They're both located on top of each kidney. So here's a kidney, and here's an adrenal gland. Here's another kidney, and here's the other adrenal gland. And then you can see it in this cadaver image right here. So the anatomy of the adrenal glands is you have this outer cortex area, so this dark outer area is called the cortex. Sometimes you'll see it called the cortical region. And then the inside, this lighter portion where you see these blood vessels, is called the medulla or the medullary region. And then there's a capsule, this beige thing on top is, so you've got the capsule, and then the cortex, and then the medulla. Okay, so the medulla area is responsible for releasing what are called catecholamines. Catecholamines are epinephrine and norepinephrine. Those are your adrenaline molecules, and those get stimulated to be released by activation of the sympathetic nervous system. So you have neurons coming into the medulla of the adrenal gland during your fight or flight response, causing it to release your adrenaline, epinephrine, and norepinephrine. The outer part, the outer cortical region here, the cortex, releases a variety of type of steroid hormones. Histologically, you can see the capsule here. This, and this are all part of the cortex. There's three different regions of the cortex: the zona glomerulosa, the zona fasciculata, and the zona reticularis. And then this down here is the adrenal medulla. So hormones in the adrenal cortex, let's talk about aldosterone. Aldosterone is a type of what's called a mineralocorticoid. It's produced within the cells of the zona glomerulosa. So again, here's our capsule. All of this region is the cortex, This top region of the cortex is called the zona glomerulosa. That's where you would find aldosterone. So what aldosterone does is it regulates the concentration of sodium and potassium in our body fluids, so our blood and our urine and other body fluids. And we can do that by regulating how much stays in the body versus how much gets excreted in the urine. Okay, so within the cortex, the top layer is the zona glomerulosa. That's where you would find your mineralocorticoids, which is a broad kind of term for a hormone that regulates electrolyte and fluid balance. Aldosterone is a specific hormone. It's a type of mineralocorticoid that comes from the zona glomerulosa. Cortisol and corticosterone are two hormones produced by the zona fasciculata. Okay, so here, the largest region of the cortex is known as the zona fasciculata. This is where we find these stress hormones, cortisol and corticosterone. These two hormones serve to increase the nutrient levels in the blood. So nutrient levels, we're talking about glucose, fatty acids, and amino acids. And that's an important thing that needs to happen during a stress response or when we're responding to something such as tissue injury. So anytime you have any of that, you will get the release of those stress hormones, cortisol, corticosterone, from the zona fasciculata. And then continuing to move down in the cortex, you have the zona reticularis, which releases something called gonadocorticoids. Gonad, that should sound familiar. So basically, what gets released from the zona reticularis are hormones that are largely released from the gonads, but we do have some release of them from the adrenal cortex. So androgen, the term androgen is a general term for all testosterone-like molecules. So we're all familiar with testosterone, but there's different varieties. There's dihydrotestosterone. There's a couple of other types of molecules that basically do the same thing. So androgens is like the collective term for testosterone-like molecules. So that's actually what gets released from the zona reticularis of that adrenal cortex. So it happens both in males and in females, but in females, we have an enzyme that converts those androgens into estrogen so that we don't become masculinized by having too high of levels of androgens or testosterone. Now, one thing that can happen if you have an adrenal tumor is that you get higher and higher levels of androgens, that we don't have enough enzyme to continually convert those to estrogens. And so you can get some level of masculinization in the female if you have like an adrenal gland tumor that's really pumping out these androgens or testosterone. So to look at the hypothalamic pituitary adrenal axis, or HPA axis, so again looking at the communication and interaction between the hypothalamus, the pituitary, and the adrenal glands. So this is the same kind of concept we looked at with the thyroid hormone. I really like these diagrams. I think they're very, you kind of have to go step by step to follow them, but it explains the whole process nicely. So the first thing that's going to happen is you have to have a stimulus. Negative feedback by cortisol, time of day, stress is probably the number one stimulus that the hypothalamus is going to respond to, and the hypothalamus is going to release corticotropin releasing hormone, or CRH. That CRH goes down to the primary capillary plexus, which then travels through that hypothalamo hypophyseal portal system, that blood system, that then takes it to the anterior pituitary and would stimulate the anterior pituitary to release ACTH. ACTH is adrenocorticotropic hormone. You can see that over here. So adrenocorticotropic hormone comes out of the anterior pituitary. It travels through the systemic circulation until it reaches its receptors on the adrenal gland. When it does that, it stimulates the release of cortisol from the adrenal gland, and then cortisol then comes out into the systemic circulation. So again, down here in the blue box is all of the effects of cortisol. So you're going to get a stimulation of gluconeogenesis. So we're going to use amino acids and fatty acids. So you can see these amino acids are coming out of the bloodstream into the liver. And we're going to use those amino acids to create glucose. So the glucose is then going to come out of the liver. Stimulation of lipolysis and inhibition of lipogenesis. So again, that reference slide, if you don't remember these terms, but if you remember, lysis is anytime we break something up, and genesis is anything that we form or create. So lipolysis, we're breaking up lipids. We're breaking up these triglycerides into glycerol and fatty acids, and those are going down into the bloodstream. And we're going to inhibit lipogenesis. We don't want to create lipids at the same time we break them down. So it's sort of intuitive there. And then all of our cells are going to undergo what's called protein catabolism. So we're going to break down some proteins and release the amino acids into the bloodstream. So these are all things, biochemical changes, metabolic changes that you would see once you get that release of cortisol coming out of the adrenal gland. So cortisol, if you remember, was one of those steroid hormones. So it gets released from the zona fasciculata, from the adrenal cortex, and because of its steroid structure, it has to travel through the blood attached to a carrier molecule. So corticosteroid binding globulin, or CBG, is the protein that kind of carries cortisol throughout the blood. Corticosterone is often used to treat inflammation because it suppresses the immune response. So one component of the immune response is the inflammatory response. And sometimes with inflammation, it kind of does more harm than good. A certain amount of inflammation is good, but oftentimes it gets kind of carried away and we get a whole bunch of inflammation, which now causes more issues. And so oftentimes we want to suppress that inflammatory response, and corticosterone and other steroid hormones like that are good for suppressing inflammation. One of the risks of doing so, however, is suppression of the immune response then can put an individual at higher risk for infection. So it's kind of like a balancing game. If you've got something that's causing some inflammation, that inflammation gets out of hand, you can suppress it. But if we're talking about cortisol and taking corticosteroid, a general suppression of the immune response or the immune system can lead to an increased chance of catching some kind of infection. Disease is associated with inappropriate release of hormones from the adrenal cortex. One of those is Cushing's syndrome. So Cushing's syndrome is characterized by obesity, specifically in the face and the back. So when it's in the face, they refer to it as moon face, and in the back, they refer to it as a buffalo hump. Osteoporosis, muscle weakness, salt retention resulting in overall swelling. Those are kind of the general characteristics of Cushing's syndrome. And this is caused by chronic exposure of body tissues to glucocorticoids. So glucocorticoids is kind of another term for corticosteroids that we talked about on the slide just before this. So when is a time that you would have chronic exposure to something like that? If you're being treated for, say, an autoimmune disease. Okay, so again, our immune system is there to protect us. It's there to, if it's working properly, it's supposed to only attack things that are harmful, foreign, things that our body aren't supposed to have. But sometimes it goes awry and your immune system starts to attack normal, healthy cells in your body that your body needs. And that's called autoimmune. It's your immune system attacking itself. Well, we need to suppress that because we need those cells and we don't want our immune system taking them out. So the only way to do that is this kind of chronic exposure to corticosteroids or glucocorticoids to suppress that immune response. And then again, that chronic suppression can lead to Cushing's syndrome. So you can kind of see the difference in this lady's features before Cushing and then after Cushing set in. On the other end of the spectrum, you have Addison's disease. So Addison's disease is when you have adrenal insufficiency. So as opposed to having chronic exposure to corticosteroids like you did in Cushing's, in Addison's disease, you don't have enough exposure to corticosteroids or glucocorticoids. So characteristics are weight loss, fatigue, hypotension, which is low blood pressure, and this kind of general darkening of the skin, so you can see before before being treated for Addison's disease versus after. You can kind of see the difference in his appearance. Again, it's caused by adrenal insufficiency, so there's an overall shortage of glucocorticoids or corticosteroids in the body. So whenever you don't have enough, you can simply use oral supplementation, you know, to take corticosteroids, but you just have to do that for the remainder of one's life. Moving on to the pancreas. So the pancreas lies posterior to the stomach, in between the duodenum, which is the very first part of the small intestine, and then the spleen. So here is the pancreas, right here. This right here is the duodenum. This is the first part of the small intestine. And over here is the spleen. And then the stomach would lie right anterior, or just in front of the pancreas. Pancreas releases the hormones insulin and glucagon. Those two hormones have opposite effects, but they're both involved in regulating blood sugar. So the pancreas is composed primarily of what is called pancreatic acidae. The cells of the pancreatic acidae, these are exocrine cells. They work in an exocrine fashion. So we know that endocrine means that we're getting something released into the bloodstream. The term exocrine means you're releasing something anywhere other than the bloodstream. So it could be the oral cavity, could be the stomach, or the GI tract, or even the surface of the skin. So specifically in this case, the majority of the cells of the pancreas, or the pancreatic acidae, work in an exocrine fashion to secrete digestive juices into the small intestine. So that its primary exocrine role is to aid with digestion. But scattered amongst those pancreatic acidae, you have these clusters of endocrine cells. These are called islet cells. The actual term is called islet of Langerhans. So pancreatic islets are composed of two major cell types. You have alpha cells. Alpha cells are the ones that are responsible for releasing glucagon. Glucagon is a hormone. And you have beta cells. Beta cells release insulin. Insulin is also a hormone. Glucagon gets released whenever your blood sugar goes down, and its job is to help it go back up. Insulin gets released when your blood sugar goes up, and its job is to help get it back down. So the primary endocrine function of the pancreas is to maintain your blood glucose level. The normal physiological range for that is going to be between 70 and 100 milligrams per deciliter. So we're constantly, varying when we eat, when we don't eat, how much we eat, how little we eat. So despite all of that, our blood sugar needs to remain between 70 and 100. So if we do eat a large meal and our blood sugar goes up, we would release insulin to help clear that blood glucose out. It moves the glucose out of the blood and into the body cells. If we don't eat for a long time and our blood sugar goes down, we get the release of glucagon. Glucagon pulls glucose out of our body's stores to get our blood sugar level back up. So here's our diagram that's going to talk about lowering blood glucose with insulin. So the stimulus here is an increase in blood glucose, increase in blood sugar. That gets detected by the endocrine cells of the pancreas. The beta cells specifically are going to release insulin. Insulin is going to go out into the bloodstream, and then it's going to cause all of the following effects that are highlighted here in the blue box. So the goal here is to get the glucose out of the blood. So in the liver, what's going to happen is you're going to have an increased glycogenesis. So remember from that reference slide, glycogenesis is when we take extra glucose that we have and string it all together to create glycogen. That's going to be our stored glucose. So we're going to have increased glycogenesis. We're going to stop glycogenolysis and gluconeogenesis. Glycogenolysis is a breakdown of glycogen, so that would increase blood sugar, so we don't want to do that because we already have high blood sugar. Gluconeogenesis also is going to create glucose. We don't want to create it because we just took it in through our diet. So primarily we're going to increase glycogenesis. In our adipose or our fat tissues, we're going to increase lipogenesis. So when we think about insulin, we always associate it with blood sugar. We move our glucose out of our blood and into our cells. But we do that for actually all of our nutrient molecules, meaning glucose, fatty acids, and amino acids. So the fatty acids that came in through the meal as well are going to be moved into the adipose tissue, and we're going to increase lipogenesis. We're going to increase the amount of triglyceride synthesis that we're doing. Also, we're going to move the amino acids out of the blood into our cells, specifically or mostly our muscle cells. So increase uptake of amino acids, and that's going to stimulate protein anabolism. We're going to use those amino acids to build new proteins. And then glucose is going to get taken up by most of the cells of the body. They have glucose transporters that allow that glucose to move simply out of the blood and then into the cells. Effects of insulin: increase glycogenesis in the liver, so we're going to take that extra glucose, create glycogen out of it, decrease gluconeogenesis and glycogenolysis. If the words on the slide get confusing, go back to that reference slide that I showed you that had all these definitions. For our adipose tissue, we're going to increase lipogenesis and decrease, this should say lipolysis right here, but you can even just cross that out and just know that we're going to increase lipogenesis. We're going to take those fatty acids out of the blood and create triglycerides from it. Increase the uptake of amino acids, specifically in our muscle tissue, to stimulate protein anabolism, or the buildup of new proteins. It's going to stimulate most other body cells to fuse vesicles containing the GLUT4 transporter to the plasma membrane. So what this is telling you is the GLUT4 transporter is the transporter that allows glucose to move out of the blood and into the cells. That GLUT4 transporter doesn't just constantly stay on the surface of our body cells, or we would be constantly sucking the glucose out of our blood. So rather, they hang out in these little vesicles right underneath the plasma membrane. And when insulin gets released in response to high blood sugar or an increase in blood sugar, that GLUT4 transporter kind of inserts itself into the plasma membrane, acting like a doorway or a gate to allow the glucose then to move out of the blood and into the cells. So overall, insulin results in a decrease in all of the nutrients in the blood, so amino acids, fatty acids, and glucose, and it promotes other cells to utilize the glucose that's available in the blood. So we're going to first take that into the cell. That is going to result in lowering our blood sugar because we're moving the glucose out of the blood into the cell, and then our cells can utilize that glucose. Okay, so what do we need to do if we need to increase the blood glucose with glucagon? So let's say that we're taking a trip in the desert and we get lost and we didn't bring enough food, and now we're really kind of starving. We haven't had enough nutritional intake. So what's going to happen is our blood glucose is going to go down. That's going to get detected specifically by the alpha cells in the pancreas, and the alpha cells are going to release glucagon. So here comes the glucagon into the circulation. And again, down here in the blue box are the effects of the glucagon. The liver is going to have increased glycogenolysis and gluconeogenesis. So glycogen is our stored glucose. If we increase glycogenolysis, we're going to start breaking down that glycogen into individual glucose molecules, and it would release glucose into the bloodstream. Gluconeogenesis is when we create glucose from non-carbohydrate sources, so we can create glucose here and then again release it into the bloodstream. And then for our fatty acids, we're going to increase lipolysis, so we're going to break down these triglycerides into glycerol and fatty acids and release those into the bloodstream. So again, in list form, effects of glucagon: increase glycogenolysis and gluconeogenesis, specifically in the liver cells. Those are called hepatocytes. Hepatocytes are the term for liver cells. And inhibit glycogenesis. We're going to increase lipolysis and inhibit lipogenesis. And then we're going to cause an overall increase in glucose, glycerol, and fatty acids in the blood. Okay, important note down here: the nervous tissue relies almost exclusively on glucose for function. So therefore, a drop below normal puts one at risk for impaired mental function and even death. So if you've ever seen a diabetic or somebody with really low blood sugar, they start to act almost like you would expect somebody to act when they're drunk or something, intoxicated, impaired. And the reason for that is because our brain tissue relies almost exclusively on glucose. And so if there is a decrease in the glucose that's made available, you really start to see some impaired mental function. Conditions related to abnormal blood glucose levels, diabetes mellitus, we're probably all somewhat familiar with this, characterized by high blood glucose levels due to inadequate uptake of glucose by the cells from the blood. So normally we eat the meal, our pancreas detects an increase in blood sugar, pancreas releases insulin, insulin causes the movement of that glucose out of the blood into the body cells. If that doesn't happen, then the blood glucose stays elevated. So if left untreated, it can cause vascular damage, blindness, and kidney damage. It can be treated with insulin injections. However, it's a little bit different than what we saw with the, disorders of cortisol or thyroid hormone. We can't just take an insulin pill once a day and be fine. And the reason for that is because we've got to keep those levels between 70 and 100 milligrams per deciliter, and that's constantly changing based on when we eat and when we don't eat. So basically what we have to do if somebody has diabetes mellitus is constantly monitor their blood glucose level. And then when it starts to go up, which is what would be expected after a meal, they've got to administer the proper amount of insulin to shuttle that glucose out of the blood into the body cells. So typically type 1 diabetics use insulin injections and type 2 diabetics take medication. This is not like a steadfast, type 1s I think also always rely on injections, but there are cases where type 2 diabetics also use injections. The main difference between type 1 and type 2, in type 1 the beta cells just completely stop producing insulin. They are just done and they're just not going to produce any more insulin. In type 2, the beta cells either don't produce enough or the cells have stopped responding to it. The cells aren't as responsive to the insulin. So there is kind of some functioning in the type 2, but there's something going wrong where it's just not functioning efficiently. In type 1, you've got a complete cessation or a complete stoppage of insulin production by the beta cells. Hypoglycemia, this is low blood sugar. So if the blood glucose level drops below 60 milligrams per deciliter, this tends to be a situational occurrence. So an insulin overdose, so let's say a diabetic ate a meal and they overestimated how much insulin they needed. So they took too much and then that insulin just wiped out all the glucose that was in the blood. That could lead to low blood sugar, hypoglycemia. Prolonged and intense exercise, alcohol consumption on an empty stomach. So these can be overcome by eating or drinking. If the person's unconscious and they can't eat or drink, you can use a glucagon injection, and that's going to stimulate the liver to start releasing the glucose from those glycogen stores.